In this video, I will be addressing the stages of mitosis, also known as the cell cycle. Before getting into the cell cycle itself, we must understand a few key concepts. Firstly, diploids versus haploids. Now, both diploids and haploids contain DNA. However, a diploid contains 2 copies of each DNA. It also uses the form of reproduction known as meiosis. On the other hand, a haploid only has 1 copy of its DNA, and it uses the form of reproduction known as mitosis. Each DNA set has 23 chromosomes. Since a diploid has 2 copies, it will have 46 chromosomes, whereas a haploid, which only has 1 copy, will have 23. Next, we must understand what a chromosome is. A chromosome carries all genetic information of the specific cell. Naturally, chromosomes are in a chromatin form in their cell. This means they are loose, making proteins and controlling the cell. However, during interphase they replicate themselves to create a sister chromatid. They are attached at the centromere, ready for mitosis. Now, let's look at the concept of mitosis. Mitosis is a form of asexual reproduction. This occurs in all body cells except egg and sperm cells. It is the process by which cells create offspring by dividing and producing 2 genetically identical nuclei. For example, if you get a cut, it will heal over time thanks to cell replication, which is mitosis. Mitosis is also part of the cell cycle. However, it only equates to 10% of the cell cycle itself. The other 90% is interphase. Interphase is a crucial stage in terms of preparing the cell before mitosis. It consists of 3 phases, G1, S, and G2. Let's start with G1. G1, standing for gap 1, is the process where the cell is prepared for division by increasing in mass. During this stage, the cell continues to perform normal functions. During the S phase, the DNA is duplicated, as well as the replication of the chromosomes and the centrosome. And finally, the G2, gap 2 phase further synthesizes proteins and continues to grow in size. After completing interphase, the cell is ready to move into mitosis. Again, mitosis is split into 4 different phases. We have prophase, metaphase, anaphase, and telophase. Prophase. During prophase, the key things that you will notice is that the nucleus itself dissolves to allow separation of the chromosomes. Next, the sister chromatids form an interphase, become tightly coiled X-shaped chromosomes. It coils so that it can easily be divided. The spindle poles begin to form at opposite ends of the cell. Spindle poles are made of long proteins called microtubules. They organize chromosomes to ensure that each daughter cell will contain 1 exact copy of the parent cell DNA. Metaphase. During metaphase, chromosomes line up in the middle along what is known as a metaphase plate. The microtubules, which make up the spindle, then attach to these chromosomes so that they are ready to be divided. Anaphase. During anaphase, the chromosomes are pulled apart and moved towards either end of the spindle. Both sides have exactly the same genetic material. The cleavage furrow begins to form so that the cell membrane is prepared to split. And finally, the last stage of mitosis, telophase. Since the original nuclei envelope was dissolved during prophase, during telophase, 2 new nuclei envelopes form around the loose chromatin, as well as a new nucleus in both sides. The cell continues to elongate, with the cleavage furrow becoming smaller and smaller. Now, there is one more stage left of the cell cycle, and that is cytokinesis. Cytokinesis is simply the division of the parental cell into the 2 daughter cells. Once they are divided, the 2 cells repeat the entire process over and over again. Wait, what?